Hi guys, sorry for the minute delay or so. My technology was being inconvenient, but welcome. This is Caroline with Fiveable. Thanks for joining in tonight. Tonight we're talking about gene expression, gene regulation, cell specialization, and mutations. So all good things. I'm not sure, I didn't see, actually, let me really quick change. Oh no, it should be okay. Hopefully you guys have access to this. If you don't, I'll do the settings, but here's the link. I don't think it went through earlier. So there's that for you guys, unless it's on there, in which case you have a choice. And I hate when this thing gets so big. Just as a reminder, here's our schedule. So we stream every Wednesday at 8 p.m. Eastern. Remember, all these times are in Eastern. So if you're in a different time zone, just convert. And everything is free now. So I'm sure you guys saw that. And if you're joining for the first time, welcome. So happy to have you. Really excited that this is a free service now. And we can talk a little bit more about it just in case this is your first time joining. But basically, I'll run through these slides. Obviously, these are three major topics, and I'm going to be talking for maybe 40 minutes. So we're not going to get into excruciating detail here like you would with your teacher that you see every single day. But the best thing that we can do here is just build off of questions that you have. So hopefully my slides spur something inside of you, a question that you might have or have been wanting to ask, and I can give you extra examples or more information. So that's kind of the goal. Coming up, mark your calendars. So next week, we're talking about microevolution and Hardy-Weinberg. Hardy-Weinberg is always a big one that people are sometimes confused about, so make sure you tune in for that. The week following will be macroevolution, and then we'll be talking about extinction, variation in populations, and origin of species. So lots of evolution topics coming up. And as always, follow us on all of our social media. That's where you'll get all of our access. We are offering a Cram pass right now. So Cram passes will get you, it's actually me, I'm streaming for bio, 10 Cram sessions. So one review of each unit, followed by some sessions leading right up to the AP exam. The last session that we'll do will be the Sunday night before your AP exam. So definitely invest in one of those if you want some extra support. And as I said, in this stream, we'll review the central dogma. If you tuned in with Jessica last week, then you should have a good understanding. So that's going to be transcription and translation. Then we'll dive into gene regulation, cell specialization, and mutations. Okay, so the central dogma. So this is our foundation. I'll go over a couple of things that you should definitely be familiar with. So just remember that we're always going from DNA to RNA to protein. And it's also important to remember that all organisms do this, okay? So whether you are a bacteria or whether you are a human, you're going through transcription and translation. So what this means is, and we'll get way more into this when we get into talking a little bit more about biotechnology, but what this means is that we can take a human gene and put it into a bacterial cell, and that bacteria will produce protein, the same protein that we would produce, because they also have DNA and RNA with the same bases, and they have the same amino acids. So we're going to talk about bacteria actually a lot today, but just important to remember that. That we actually do this all the time. So insulin, which is needed to help regulate your blood sugar, and it's something that people who have diabetes are struggling to regulate, that we can actually make by putting the gene that codes for proteins that create insulin into bacterial cells and having them produce that. So just a fun fact there for you. That's biotechnology. But again, it's because they're going through these same processes that we are, using the same materials. So on the left-hand side, we have transcription. Just a reminder, we're going from DNA to RNA. Sometimes students get transcription and RNA or DNA replication confused. They are similar processes, but with some key differences. So RNA polymerase is used here. So remember, anything that ends in ACE is an enzyme. ACE is enzyme. So this is the enzyme that makes the polymer RNA. And so what they're doing here is the DNA is unzipping. RNA polymerase is going ahead and reading the open strands and putting the corresponding RNA base with that. Another thing to remember is that if that RNA polymerase is moving down the line and they detect an adenine, they're not going to replace it with a thymine because there are no thymines in RNA. They will instead replace that with a uracil. And you can see that. Let me get my pointer up. You can see that right here where we have adenine, which are these little red guys, And they're pairing with these orange uracils instead of the green thymines, which they were originally paired to. And again, that's because our RNA strand does not have thymine in it. Okay, so you'll notice in this diagram then we have this released, it's called pre-mRNA. So this is a difference between, and I'm sure Jessica went over this last week, but this is a difference between eukaryotes and prokaryotes. Prokaryotes do not process their mRNA at all. Eukaryotes do. So we'll go ahead and actually cut out regions of this mRNA called introns. So the introns stay in the nucleus and the exons exit the nucleus as part of the message. So we'll cut out those introns and then we'll add a poly-A tail, meaning many A's, many adenines at the end, and then a 5' methyl cap on the 5' end. And you can see the primes listed here. That little, if you're not familiar, that little apostrophe we refer to as a prime. So the 5' end will get a methyl cap, and that's kind of like Rudolph that guides our pre-mRNA to the ribosome so that it can bind and be translated. So I know that was a super brief overview. Hopefully, again, this is all review. Hopefully you've learned about transcription before, but this kind of APifies it when we start talking about this processing and how eukaryotes process their mRNA and prokaryotes don't. I have seen an AP free response question about that before, where they talk about how eukaryotic mRNA gets shorter before it leaves the nucleus because we're removing those introns, whereas prokaryotic mRNA does not change length because it's not being processed. Okay, and then on the right-hand side, you see translation. So this is where our mRNA joins up with our ribosome, and our tRNAs are going to come and read our mRNA in triplet codons. Okay, so that's going to be important when we talk about mutations, that triplet codon that is going to be read. And whatever tRNA has the corresponding anticodon that matches up with the codon, has a specific amino acid, and I'm sure you guys have looked at those amino acid charts before that tell you which 3 codons lead, or which 3 bases lead to which amino acid. And then that's how our specific protein is born. And I like how they say newly born protein because it sounds way fancier than it is. And then that polypeptide will get folded into its 3D shape to make the protein. So that is those 2 processes. I just wanted to review that because all of this is really, really important when we talk about how genes are being expressed and regulated. And it's also going to be important when we talk about mutations. So any questions about either of those processes before we move on? And in case you're not familiar, if this is your first time joining us, you can write a question in the chat on the right where it says say something nice, or you can put ask a question and then ask a question there. Either one will do. Sometimes I'm jumping back and forth between my PowerPoint and my Crowdcast screen. So if that happens, just I'll answer your question when I pop back over. So thank you for your patience. Jenry, I see, Henry, maybe, sorry if I mispronounce your names. What's the difference between the A site and the P site? So that's a great question. So technically there are 3 sites on the ribosome, the A site, the P site, and the E site. And so they go in that order. So A is for attachment, P is polypeptide, and then E is exit. So the A site is where, and so when mRNA is moving through the ribosome, it's kind of like a conveyor belt. It's being pulled. So as it's being pulled, right, the A site will open up and there will be 3 bases waiting to be connected with a tRNA. And those 2 things will attach to one another at the A site, okay? Then they will be pulled through and they'll move over to the second site, which is the P site. Again, picture kind of like riding a roller coaster or getting pulled down a conveyor belt. When they get to the P site, so in this example, when this lovely tRNA moves over to the P site, this new tRNA can join the A site, and then we're going to form a polypeptide bond. So that's what happens at the P site. This guy will join the growing polypeptide chain as this guy enters the A site. Okay, so this binds with a polypeptide bond. This guy enters the A site and attaches, and then this guy will move to the exit site, which just means it's going to move on. So you can kind of follow these arrows. They're showing you the movement of the blue binding and then moving away. So A for attachment, P for polypeptide bond, and then E for exit. Great question. Okay, cool. All right, so we're going to jump into gene regulation then. So what's the purpose? Why do we regulate our genes? What does this even mean? Well, the easiest way to put it is that in any of our cells at any given time, we are not expressing all of our genes, right? That would be insane. We have so many genes, and some of our cells will never ever express a gene, right? So like our skin cells are not going to be expressing the same genes as our heart cells, right? Or as our muscle cells or as our blood cells. Even though they all have the same DNA, they're not all going to express the same genes because they all have different functions. Okay. Even two skin cells are not going to be producing the same genes at the same, or producing the same proteins at the same time if they don't need to, because we want to save energy. So we only want to turn on certain genes at certain times, right? If we need them. Because again, if we were transcribing and translating every gene that we have at all times, we wouldn't survive because it would just be too much energy. So second bullet point then, not all genes are used at once, as I said, and not all genes will be used by a cell in its lifetime, right? If you're a heart cell, then there are some genes that you will never express because your job is to be a heart cell. Even though you have those genes, you will never actually express them. And when I say express them, I mean transcribe and translate them into protein. DNA itself doesn't really do anything. It just sits in the nucleus. It just codes for other things, right? It's the proteins that it codes for that are the actual doers of the cell. So that's kind of another important thing. I'm just like going to go on little tangents where I give you advice of what you should be including in free responses. You should always be writing about the protein that's doing something, not the gene itself, right? So that sometimes students get kind of confused by that. But the DNA itself doesn't do anything, right? It's just a code. Okay, so when we're regulating these pathways, there's a couple different ways that we can do this. And we will talk more when we get into different homeostasis things about like positive and negative regulation. But here, when we're talking about regulating, we're going to get some sort of signal. And this is the same as any kind of signal pathway. We're just expressing something a little bit differently. But we'll get some sort of signal. It's going to trigger a gene regulatory network component, which is a really fancy word. We're going to look at some examples of that, like repressors and activators, something that's going to do something to regulate a gene, and then that's going to lead to primary and terminal outputs. So a primary output would be changed RNA and protein complements, and then terminal would be an overall change in behavior or structure, okay? So to simplify this, basically we get a signal, we decide what we're gonna do with that signal, and we either increase or decrease the production of a specific gene, which leads to a specific protein, which does a specific thing. So that's like the most basic way to say this, right? But these are all triggered by something. And we're going to look at two different pathways, one that is normally active and one that is normally inactive, and we can talk about some examples of what that might look like in different cells, okay? But we have a stimulus that's leading to some sort of regulation, either upregulation or downregulation, which is going to lead to or not lead to the production of a protein. And you can see that there's a bunch of arrows going back here too, because there's negative feedback coming back and saying, hey, we don't need this thing anymore. Like if we're supposed to be making a particular protein, we may only need that for a short period of time, and then that protein may actually inhibit its own synthesis. And that's something that we'll talk about when we talk about inducible operons, but I'm getting ahead of myself. On the right-hand side, just another way to look at this. This is just one other example of gene regulation. This is hormone gene regulation. So you'll notice that the hormone goes right through the plasma membrane, tying everything together here. This is probably a steroid hormone. They are nonpolar and can get through the membrane, so that they can therefore activate all cells. And they're binding with some sort of complex that's leading to the transcription and translation of a new protein. So when this hormone enters the cell, a protein is being translated. And that can be any number of hormones and any number of proteins that would lead to that. But that's what we're really trying to show here. This protein would not be made unless this protein was activated, this receptor protein was activated by a hormone. So hopefully this general overview kind of makes sense, because we'll get into some more specifics. Okay, so we're going to be talking mainly about prokaryotic gene regulation. Why? Because it's a lot simpler. It's a lot simpler for me. Whoa. Sorry, guys. It's a lot simpler for me to explain to you how prokaryotes regulate themselves versus how we regulate ourselves, because we are very complicated. So we will only look at us a little bit. It's more important to me that you guys understand prokaryotic gene regulation and the two different types. So we have repressible and inducible operons. So an operon in and of itself is just a unit, okay? It has a promoter, it has an operator, and it has genes. And I'm going to define all those things for you on the next slides. The promoter is going to be where RNA polymerase binds. The operator is going to be like our switch. So that's where a repressor may or may not be bound. And then there's a set of genes that correspond to this. So think about a light switch. You can turn a light switch on, and it may turn on a lot of different lights in your house. So let's say you flip a switch in your kitchen. You may have 5 lights in your kitchen, 5 overhead lights. Think about that, right? So you're switching that light, and all 5 of them go on at the same time. You don't have to go and individually like pull the cord for each of those lights, right? Back in the day, you may have had to. So this example then, we have one switch, this operator that controls all of these genes. In this case, it's the 5 genes that we see here that are going to be involved in making tryptophan. But with just one switch on or one switch off, we can control this entire set of genes. So that's another way that we're saving energy. Sorry guys, when I talk sometimes it makes me yawn. Okay, so let's look at some key vocabulary before we jump back in, and we'll take a look at that same operon and then a different version. So good things to know. So an operon is the entire stretch of DNA required for the production of a certain protein or set of proteins, depending. Oftentimes it's a set of proteins that will do the same function. The operator is the switch, as I said. The repressor is the protein that switches off a gene. So that is going to repress, right? So we can see that here in red, or stop the movement. And I always think of the repressor as like a tree in the road. Okay, so if RNA polymerase is our car and this repressor is like a tree in the road across, we're not getting across that tree. Okay, so this RNA polymerase then can't find these genes down here in order to transcribe them, which would be the first step in making this into a protein. Okay, so if we remove this tree like we have here, then our RNA polymerase can go through and we're able to transcribe and translate. Okay. The regulatory gene. So the regulatory gene I actually don't have a picture of for you, but this is a different gene. It's going to be upstream. And by upstream, I mean before. So it's going to be before our actual operon. Okay. And it's going to make the repressor protein. So it's responsible for making that protein because somebody's got to. There's got to be genes somewhere that code for it. And that's going to be the job of that repressor. And then we may sometimes see a co-repressor. So a co-repressor would be a molecule that binds to a repressor to activate it. So for example, okay, here, this repressor is inactive, right? We can tell it's inactive because it's not bound to our switch or our operator, okay? When tryptophan binds to our repressor, this would be considered our co-repressor, okay? So like a co-captain, it helps this repressor to be active. So when these two things are together, they're capable of binding to the switch and blocking RNA polymerase, okay? So that is the job of a co-repressor. Some have it, some don't. We'll see an example of both. And then lastly, our promoter, as I said, is the region in which RNA polymerase binds. So those are all some important vocabulary that will make more sense as we're moving through this. Do you have to memorize all of these things? Probably not, but it's good to be familiar with them in case you need to put them into context. I have seen something kind of similar to this before, and I actually have an example of a free response question at the end of this that we can take a look at. Okay, so there are two types of operons, which is kind of what I've been alluding to. We have repressible and inducible. So repressible means it is able to be repressed. Okay, what does it mean to be repressed? It means turned off. It means inactive. Okay, so a repressible operon is capable of being repressed. Okay, if you are able to turn it off, think about the light again. Okay. If you are able to turn it off, that means that it's on. It is naturally on, okay? Think about that. Just process that for a second because these can be kind of confusing. But instead of just having to memorize these things, if you actually think about the word, it can make a lot more sense. So inducible, on the other hand, means able to be or capable of being induced. What does it mean to induce something? You can induce labor. It means to turn on or activate. Okay, so if this thing is able to be turned on, that means its natural state must be off, right? You are able to turn on, I'm trying to think of like a better example than a light, but I guess a light is a good example. Maybe repressible would be like your phone, because you never really turn your phone off. It's naturally on, okay? But you could turn it off if you needed to, okay? Versus a light, which might naturally be off, like your bedroom light is naturally off, but you're able to turn it on when you come in there and you need light, okay? So there's an example for you. But you can also read through these, and when we go through each of these in more in-depth detail, it should make a little bit more sense. Okay, so I should have flipped these orders because I explained them in different orders, but that's okay. So we'll start with this inducible operon. So remember, inducible means able to be induced or able to be turned on. Okay, so what that means is this guy is naturally not on. Okay, and we can see that up here. So here's our regulator gene. So I do have a little pickup. They call it Regelin, Regelgen. But here's our regulator genes making mRNA, turning into this repressor protein that's binding to the operator. So you'll notice right off the bat, this would just be a normal, like a day in the life, okay? Remember, these are bacteria, so this would be the normal repressor that's made in a bacteria. So this guy is normally off because there's a tree in the road, right? This operator is blocked, RNA polymerase can't get through, and therefore we can't make any of the proteins after this. So this inducible operon is known as the Lac Operon, LAC Operon, okay? Lac Operon, oops. And it is responsible for producing proteins that break down lactose. So you can see the different fancy names, beta-galactosidase, permease, and transacetylase. Those are 3 enzymes that help to break down lactose, which is a sugar that's naturally found in milk. Okay, so it's actually a pretty complex sugar, much more complex than like glucose or other sugars that we have, sucrose. Okay, so when lactose is present in the environment of a bacteria, they're going to break it down. But on a normal day, those bacteria may not come into contact with lactose, and therefore we don't want to waste energy making these proteins to break down something that we don't even have. Okay, why are we going to make this product if we don't even have anything to break down with it? Okay. So when this guy is going to turn on is when lactose is present. That lactose is our inducer molecule. Okay, so let's say these are E. coli that live in your intestines. We have trillions of bacteria in our intestines. We actually have more bacterial cells in us than human cells. And let's say normally you don't really eat cheese or milk or dairy products, but one night you have a milkshake. Okay, so normally the E. coli that live in your digestive tract aren't making these proteins because they have no need. But on this night, you have a milkshake, okay? So there's tons and tons of lactose that needs to be broken down by the E. coli in your intestines, okay? So the lactose molecule will bind to the repressor. And when that happens, which you can see here, it distorts that repressor. And now this repressor is no longer capable of binding. You can see its wings are kind of out. It's no longer capable of binding into the operator region, which leaves RNA polymerase free to make these enzymes to break down the lactose that you have consumed. Okay, so when lactose is present, this thing is turned on. When lactose is not present, it's turned off. Okay, once all the lactose is broken down, the inducer will be removed and we'll turn this guy back off. So it's a very good regulating switch. And we only turn this on when the substance, such as lactose, is present. Any questions on that? That was a lot of information, and I know I'm talking kind of quickly. So definitely ask questions if you have them. That is the beauty of a live stream. Anything? No? Okay, I'll take your word for it. I don't think I'm that good at explaining, so. All right, so then the opposite of this is this repressible operon. So again, remember, repressible means able to be repressed or able to be turned off, okay, which means it's normally on, okay? So our example of this, and again, this is in bacterial cells, is the Trp operon, so TRP, Trp Operon, and TRP is for tryptophan. Okay, so tryptophan is one of our amino acids, okay, and it is essential that we have this amino acid because we're constantly making proteins. Remember that amino acids are the building blocks of protein, okay? So we always need, and bacteria always need, a steady stream of tryptophan in order to make protein. The problem is tryptophan is not naturally occurring that frequently. It's in things like turkey, but it's not a huge part of our natural diet. What that means is we are going to have to go ahead and make our own tryptophan in order to make sure that we have enough to make protein. So with that being said, tryptophan is something that we need to survive always. So instead of being an inducible operon, this is a repressible operon, meaning naturally, right, when we don't have any tryptophan, our regulatory gene creates a repressor that's inactive. So naturally, when nothing else is present, this guy is on and activated. The RNA polymerase is able to transcribe and transcribe these stretches of gene, which will then be translated into protein, which will then create the molecule or the amino acid tryptophan. Okay, and that tryptophan then can be used in a number of proteins. Okay, if for whatever reason we've made too much tryptophan, or you're an E. coli in our stomach again and it's Thanksgiving and we eat a bunch of turkey and there's a whole surplus of tryptophan, we don't want to waste our energy making this thing that we actually have in our environment. So in that instance, the tryptophan, right, being in surplus, binds to the repressor, and that binds to this operator region and prevents us from making any more tryptophan. So that makes sense. We don't want to make something that we already have plenty of. Okay, so it kind of turns itself off when we've either made too much or come into a natural supply of the tryptophan. Okay, these two bind together, bind to the operator, and we're good to go. Okay. So the big difference there, the way that I always think about this, is the repressible operon is going to be used for something that we can't live without, okay? Because it's naturally on. It's only going to be turned off if we have too much of something. On the other hand, an inducible operon is going to be for something that we only have occasionally, okay? So something like lactose, we want to turn this on when it's present, but off when it's not so that we can save energy. That kind of goes back to like the phone versus light example. Your phone you want on because you need it to live. You need it to survive. You need it with you at all times, versus the light in your bedroom, which you only need maybe an hour a day, right? And then it goes off because you're either asleep or you're not in your bedroom, okay? So just an example there of when these two different operons would be used. Okay, eukaryotic gene regulation. As I said, I will go into very little detail about. I just want you to know that it is immensely more complicated than prokaryotic gene regulation. And that's probably not surprising to you. There's a really good image from a textbook that I'm not technically allowed to use, but maybe I'll post the link into the chat box. Let me just, gene regulation, eukaryotes. Let me just see if I can find it for you guys because it shows the different levels. Yeah, well, kind of. Okay. Here's an example that you guys could take a peek at. But we're going to regulate at a bunch of different levels, okay, because our genes are much more complicated and we have a lot more of them. So I've just highlighted two here for you. So on the left, this is kind of how we regulate our genes at transcription, kind of similar to what prokaryotes do. We are going to mainly regulate our genes at transcription. And if you think about it, think about the process of going from gene to protein. The very first step is transcription. So if we are trying to make or not make something, it is best to decide right away. Because if we make the entire protein and then we decide we don't want it, we've wasted a lot of energy and now we have to break down this protein and start the whole process over again. So if we can just decide not to make it in the very beginning, that's going to be the most beneficial. So us, just like prokaryotes, are going to regulate primarily at the level of transcription, okay? Because it's the first step. So what you can see here is that our DNA actually bends in a very complex way when we're going to go ahead and transcribe something. So our transcription factors are actually allowing this DNA to bend on itself. And you don't need to know any of these things in detail, but basically that's what puts the RNA polymerase on the promoter, because we have all of these proteins binding to other parts of DNA called activator regions that are way far away from this, that are forming this entire conglomeration, which we call transcription factors and mediator proteins, which are allowing RNA polymerase to move. Okay, so there are a lot of moving factors here, right, that involve the bending of this DNA, regions very far upstream of this, and a bunch of different proteins coming together in order to activate. Again, moral of the story, more complicated. Okay. The other thing that I wanted to show you is that we can actually regulate based on the fact that our DNA is wrapped really, really tightly around these little proteins called histones, kind of like spool wrap or thread wrapped around a spool. Okay. And so if that DNA is wrapped really, really tightly, you can see this pink gene here. This gene can't get transcribed or translated because it's wrapped tightly around a protein. We would actually have to unwrap this region of DNA, and you can see that there's little chemicals that are attaching here that are allowing us to unwrap this region of DNA in order to actually transcribe and translate that gene. So that's another way at this level of transcription that we can kind of regulate what genes are active and what aren't. So if you're a skin cell and you never need to express a lot of genes that would be related to muscle contraction, then those would just be wrapped tightly around histones for the entirety of your skin cell life, okay? Because there's no point in you having those. Okay, so that's all I'm going to talk about eukaryotic. I did share that slide with you. It's a picture that you can take a look at. There's a lot of different levels. It's very, very complex, but we still most commonly regulate at the level of transcription as well. Okay, so just briefly about cell specialization, and then we'll jump into mutation. As I've already said a lot today, all cells have the same DNA. That sounds really, really straightforward, but students get confused when I actually ask them, like, does your skin cell and your brain cell have the same DNA? They're like, oh, wait, I don't know. And the answer is yes, it does, right? They're just expressing different regions of it. But how that happens comes from these stem cells. So we have they call it totipotent, which is like all creating stem cells that are giving way to what we call the pluripotent embryonic stem cells. And this happens in very early development. So you only have totipotent embryonic stem cells when you are a little baby embryo of like 8 cells. Okay, and then they divide into these stem cells, the endoderm, mesoderm, and ectoderm line, that are going to create the different regions of you. So endoderm will create some inside regions, meso is kind of middle, and ecto is the outer regions of you. And those these cells can actually differentiate and specialize into all the different cells of the human body. Otherwise, a red blood cell will replicate into a red blood cell. It actually doesn't even replicate. That happens in the bone marrow. But a skin cell will replicate into another skin cell. A pancreas cell will replicate into another pancreas cell. A pancreas cell can't become a heart cell or a lung cell. It can only become a pancreas cell. So the only cells that can become different things within our body are these stem cells. And you can see there's lots of fancy names for them, of which you don't need to know all the specifics. Just know that those stem cells are the only ones that can divide into these different types of cells. Once you have a mature adult cell, those things are just going to replicate into other versions of the same cell. Okay. All right, so mutations. Mutations are kind of basic things. You've probably learned it in like an intro to bio class, but we just want to refresh your memory. So mutations, we have point mutations and then frame shift mutations. So a point mutation is just one single base changing. And when one single base changes, there can be 3 possible outcomes. The first is the best, and it's just that it has no effect on the protein sequence. So even though here we're changing from an A, TA, GTA to a GTC, this codon GTA codes for valine, and this codon GTT also codes for valine. So if you remember, when you look at the chart of amino acids, it's redundant. There are multiple sequences that code for the same amino acid. This is one example of that. So GTA and GTT both code for the same amino acid. Therefore, this protein would be exactly the same, even though there is a change in the actual codon. So that's the best kind, and we call it silent because we don't see it, we don't hear it, because it has no effect on the protein. Next one is called missense, and missense results in an amino acid substitution. So here, instead of CCC, which codes for proline, we have ACC, which codes for threonine. Okay, so that codes for a different amino acid. Sometimes that has a very small effect. Sometimes it has hardly any effect at all. Sometimes it has a very large effect, and we're going to talk about sickle cell anemia in a second. Sickle cell anemia is caused by a single missense mutation. So one single base changes, and it leads to all of these red blood cells then having this half moon shape as opposed to the round shape. So that's crazy because it's truly just one base changing. And then of point mutations, the most detrimental is going to be the nonsense mutation because it accidentally turns into a stop codon. So these are all DNA. I don't think I said that in there. Should have said that earlier, but TAC would code for AUG in, what am I saying? Yeah, AUG in mRNA, which codes for tyramine. But instead, we're changing it to TAG, just that last base changes, and that creates a stop codon instead. And that stop codon is therefore going to create a stop in our protein, and that protein will just not replicate or will not grow any further. So it's basically a dead protein, and therefore that is a very bad mutation because we're not making any product then. So that has the most detrimental effect of those three. Obviously, silent is the best, and then missense can kind of go either way. Sometimes it's not a big deal, and sometimes it is. But when you're stopping that protein from being synthesized, that's a big problem. All right, and then frame shift mutation. So frame shift is a little different. So if you remember, we were talking about reading in bases of 3. So 3, 3, 3, right? If we insert or delete any base, that is going to shift our entire reading frame. Okay, so for example, this stupid thing's going to be in my way. I'm just going to show you up here because this thing is going to block me every time my mouse goes by. If we had AGC, okay, and that goes for serine down here. If we accidentally delete this C, okay, every amino acid after this point is going to change, okay? Because now we would be reading AGG, and that's a different amino acid. And then next we would be reading TAC, which is a different amino acid, CCT, and so on and so forth. Because we deleted that base, everything shifts up to fill in that gap, and every single reading frame then is shifted with different bases in it than what was originally in there. Okay, same thing happens if we insert a new base, right? If we inserted something into the spot, let's say we insert a T there, then we have AGT, and then we would be reading CGT, ACC, and you can see that everything shifts. Okay, so with the frame shift mutation, every amino acid after the insertion or the deletion is going to change. And hopefully this is just a review. Okay, so I did mention, so sometimes students forget that mutations can actually be really beneficial. I mean, most of the change that we see is because of a mutation, right? That's why we have variation, and variation is the key to life, all those things. Did want to talk briefly about sickle cell. You can see its inheritance pattern here, but the reason that sickle cell is so prevalent, especially in places like Africa, is because if you are heterozygous for sickle cell, so you can see the two parents and then the two children in the middle that have one S and one A copy, you are immune to malaria. So malaria is a disease that you get from mosquitoes. We do not have it in the United States, but it is relatively prevalent in Africa, and you can die from it. So you can die from sickle cell, but you can also die or get really sick from malaria. But if you're heterozygous, you don't have sickle cell because you have enough regular blood cells that you're getting enough oxygen, okay? And you also can't get malaria, so you are like the supreme being. We call that a heterozygous advantage, and we'll talk more about that when we get into evolution. But because these heterozygotes have an advantage, they unfortunately are carrying that allele with them that they can then pass on to a child who could then contract sickle cell anemia, which is why we still see this so much today, just because there are a lot of carriers out there that are reproducing with other carriers. So it is very beneficial if you're heterozygous, obviously not beneficial if you are fully infected because it can be life-threatening. Thankfully, in the United States, we have good healthcare that can prevent that. But any other mutation that gives you an advantage to survival and reproduction would be a beneficial mutation. So they're not all bad. Sometimes really good things come out of it. So the last thing I wanted to go over with you guys before we leave is this really challenging free response practice. So let me just really quick, I want you to read through this question. So it's question 2. I'm trying to remember what year. So while you're reading that, I'm going to pull it up because I also want to show you the scoring guideline. Okay, so this is 2016, question 2. So if you're not familiar, the way that the AP exam used to be is you had 2 long and 6 short free response questions. Now you have 2 long and 4 short, so that's great news, but your long questions are going to be worth 10 points each, so they're kind of heavy hitters. So in this question, we have bacteria. We have 2 different axes here. So we have a bacterial population density, and then we also have a concentration of nutrients. So bacterial population, we're using the left-hand side, and then these 2 nutrients, we're using the right-hand side, and then they're asking us about these different things. So the question I wanted to show you about, or to tell you about is part D, okay? Describe how nutrient 1 most likely regulates the genes for metabolism of nutrient 1 and the genes for metabolism of nutrient 2, and provide 2 reasons that the population does not grow between hours 5 and 6. Okay, so you can look through the rest of this, and I suggest that you do because all of these are released. I'm going to post the answers for you because those are all released as well. Okay, but this whole question revolves around what we were talking about with gene regulation, and a lot of students didn't catch that because it doesn't specifically say that. So how does 1 regulate genes for metabolism of 1 and 2? Okay, so if we go back and take a look at this, we can see that nutrient 1 is being used up first. So what the College Board wanted you to say is that nutrient 1 promotes expression of genes required for metabolism of nutrient 1, and then nutrient 1 represses expression of genes required for metabolism of nutrient 2. So it's actually very simple. Nutrient 1 is stimulating its own enzymes, and it is repressing the enzymes to break down nutrient 2, and they tell you that you need to talk about both nutrients, so you know you need to talk about both nutrients, right? And if they're asking you how it works with nutrient 2, and you see that nutrient 2 isn't broken down until nutrient 1 is gone, then you can make the assumption that it is indeed repressing while it is activating its own enzymes. So it's doing both a repressible and inducible there. And then the reasons why the population does not grow between hours 5 and 6, why we see that kind of plateau, you could say that nutrient 1 is depleted or is being consumed. And then you could say that it takes time then to produce the enzymes required to metabolize nutrient 2. Okay, so we have kind of this lag phase when nutrient 1 is depleted, right? So we don't have that nutrient anymore, and then we're going to need to remove those repressors and start making the enzymes to break down nutrient 2. So just kind of an interesting, I like this question. A lot of my students missed it. That's why I wanted to put it on here. And obviously you can see the answers because I just posted them for you, but just kind of an interesting way to show this regulation that looks a little bit different. Any questions about this or any questions at all? Because this is all I have. Anything? Okay, well, if you guys feel confident, I'm excited for you because gene regulation is hard. So if you feel good about it, that makes me happy. If you have any questions, just let me know. You can shoot me a chat in the box or something like that. Would nutrient 2 suppress nutrient 1, or did it just run out? Nutrient 2 does not suppress nutrient 1. So nutrient 1 is used up first, and then nutrient 2 just runs out. So we can see that based on the relative nutrient concentration on the right-hand side that we go all the way down to 0. So nutrient 2 would not be involved in nutrient 1's regulation. Good question. Any other questions before we go? Okay. And a real life example of that would be like we always use something like glucose before we would use a sugar like lactose, just because it takes more energy to break it down. All right, y'all. Well, if you have any more questions, you can type them in the box and I'll type back to you, or I can email you or something like that. But thank you for spending your Wednesday evening with me. Join in next week for Microevolution and Hardy Weinberg, and hopefully you got some questions answered. Bye, guys.